And I'm so excited to have the start of our seminar series, our FishCAST seminar series for 2025. Just to give you a little bit of background, I'm Tina Semenuk. For those of you who don't know who I am, I'm the director of FishCAST, which is an NSERC Create program, which is all about funding the next generation of scientists. At the time when we got our FishCAST, we were the only ones to offer added value training in freshwater fisheries and aquatic sciences for our next generations. And so we are now entering into our final year of FishCAST. And one of the really important things we had decided on collectively, and I'm just so happy I'm joined by one of our amazing FishCAST PIs, Dr. Margaret Docker, and of course our incredible program coordinator, Kendra Thompson-Kumar, without whom we wouldn't have FishCAST. But we're a part of 6 different universities from Manitoba, Ontario, and Quebec. I'm situated in Wawaitanong, which is an Anishinaabe-Moan for where the river bends, which is Detroit River, where it's a little rainy right now. And I'm just so privileged and fortunate to be working on the Three Fires Confederacy of the territories of the Ojibwa, the Odawa, and the Potawatomi. And I'm so grateful to be working with every one of you here. It's been quite a journey, and it's not my journey right now to tell, but it's for our alumni to tell. So this is the second time now that we're in our stage of FishCAST to welcome back FishCAST trainees that have gone through the program. And we had an amazing seminar series last time. I'm not sure if some of you might have been on it or not just as the audience members, but I love to see what changes were done in people's careers, what they're doing now. And I'm just so pleased to have Katrina Audet with us today. But I'm actually going to turn it over to Dr. Docker to be able to introduce our esteemed speaker, where I really can't wait to hear about your journey, Katrina, and where you are. But I do want to say I was on Katrina's committee from the University of Manitoba, where she did her master's, and I couldn't believe just your fortitude, perseverance, and my goodness, the amount of detail you put into your statistics. Incredible. So I will leave it there, and I welcome Dr. Docker to introduce Katrina to you all. So thanks, everyone. Merci. Miigwech.
Yes, thank you. Yeah, it's such a pleasure to be able to introduce Katrina today. That I first got to know Katrina when she took an undergraduate class with me in 2019, and I actually had to look that up this morning because I didn't think it could possibly have been that long. But yes, indeed, I first got to know her in 2019, and then she took 2 more undergraduate classes with me, and then if that wasn't enough, that she then did a summer undergraduate research assistant placement in my lab with funding from the University of Manitoba Faculty of Science USRA program, but also through FishCAST. So this was her first introduction to FishCAST in the summer of 2021. And then she started her master's with me in January of 2022, co-supervised by Dr. Caleb Hassler. So during that time, she was always an active participant in FishCAST. As Dr. Semenuk indicated, that she was on Katrina's committee, and that connection was made through FishCAST. And she received an NSERC CGSM. I'm still getting used to the new name for that scholarship for her work comparing eDNA and gill netting to assess the presence and relative abundance of fish species in Lake Winnipeg. Very impressively, Katrina already had her job with Prairie Scientific lined up before she even graduated. And in fact, she somehow managed to finish her thesis while starting her new job. And it wasn't unusual to be getting an email often late at night that contained both the next draft of her thesis and pictures of her in the field holding a lake trout or other fish. So that, yeah, very dedicated to completing her thesis, very high quality, while also starting a new job. And I'm sure she's going to be telling us about that as she talks today. And I'm really looking forward to that. And it's my pleasure to now turn over the floor to Katrina.
Thank you, Margaret, for that amazing introduction. Okay, I'm going to share my screen here. Okay, so hi, everyone. Today I wanted to talk to all of you a little bit about my journey from my undergraduate degree and how it directed me towards my master's and then now my career in environmental DNA. So as Margaret mentioned, I'm going to repeat a little bit, but Margaret, you summarized it quite well. So I did my Bachelor of Science at the University of Manitoba in the Biological Sciences program with a focus on evolution and biodiversity. When I started taking courses, there seemed to be a little bit of a pattern that started forming, and this was taking a lot of courses about fish. And honestly, when I first started, I didn't have an interest at all in fish. To be honest, I wanted to become a doctor at the time when I first started. But there's a specific professor, Margaret, who really steered me towards learning about fish. And so I took some of her courses, chordates and biology of fishes, which really fascinated me. And Margaret's excitement for teaching about fish, I guess you could say, really hooked me onto the topic. But after biology of fishes, she invited me to join her eDNA special topics course, which ultimately influenced my decision for my master's project. And so I did my Master's of Science, again, at the University of Manitoba in the Biological Sciences department. My project was about making comparisons for Lake Winnipeg fish stock monitoring using eDNA techniques versus more traditional fish monitoring such as gill netting. Now, unsurprisingly, I had Dr. Margaret Docker as my supervisor, as well as Dr. Caleb Hassler from the University of Winnipeg as a co-supervisor for his expertise on fish stocks in Lake Winnipeg. Through the FishCAST program, I had an internship with Jeff Klein, which was really amazing. He's from the Manitoba government's fisheries department, where I got to learn and even experience myself how fish stocks in Lake Winnipeg are actually monitored. So that was super cool. I actually got to go on the boat and get hands-on experience gill netting. So that was cool. But before I move on, I do want to thank all the organizations that provided funding or made contributions in any way to my project. So FishCAST, of course, the Manitoba Habitat Heritage Corporation, University of Manitoba, specifically the Faculty of Science, GenFish, NSERC, Manitoba government, and the Lake Winnipeg Research Consortium. I also want to thank some of my committee members. So Tina, thank you so much, and Dr. Darren Gillis, as well as my colleagues like Brooklyn Litke and Jesse Ogden for all their help throughout my project. It was much needed, as Margaret mentioned. I did start my career as I was finishing up my master's, so they were all a huge help. Okay, so I mentioned environmental DNA, but what is it exactly? So eDNA is DNA that's been shed by an organism into its environment, and it can be in the shape of things like skin, mucus, excrement, or gametes. My project specifically focused on fish, and so collecting a water sample just made the most sense. But depending on the species you're looking for, you can really collect like any kind of medium from the environment. You can collect sediment samples, or you can even collect air samples. But what can that eDNA sample be used for? So this technique is especially useful for detecting species presence, mapping their distribution, especially those that tend to migrate, but also to detect more rare or invasive species. So here are a couple of the pros and cons of the eDNA technique that I make comparisons to more traditional fish monitoring techniques, things like gill netting, seining, or trapping. So eDNA tends to be less invasive because you don't actually physically need to catch the fish to say that they were present in your sampling location. And due to the nature of the eDNA technique, it is super sensitive. You really only need like one strand of DNA to be able to detect a species. And compared to more traditional techniques, eDNA has less selectivity bias. And what I mean by this is that, for example, with gill netting, smaller bodied species are often missed by this technique because of the mesh size that you use, or even the position in the water column that you place that gill net. And so eDNA tends to bypass kind of some of those biases that more traditional techniques have. But there are some downsides to eDNA. It doesn't tell you anything about the fish's health, their sex, age, size, or even how many individuals were there at your sampling location. And because of its sensitivity, there's a really high risk of contamination, whether that can be introduced through the equipment you're using from previous sampling events or even yourself. And lastly, there's a lot of factors that can influence how much DNA is actually at your site or the way that it'll accumulate in the environment. And this can be things like pH, temperature, microbial activity, how fast an individual is shedding their DNA, and so many more factors. Okay, so the traditional technique that I compared to eDNA for my project was gill netting, which are these long nets. I have a picture here. I kind of just created that on Canva just to give you an idea of how that setup kind of looked like during my project. So the net sits at the top, near the top of the water column, and fish just passively get trapped into those nets. And this technique is used every year in Lake Winnipeg through a fish monitoring program run by the Manitoba Fisheries Branch. And there are some gaps with this program, as I mentioned, because there's that selectivity bias. So larger bodied species like walleye or sauger, they tend to be caught at a much higher rate than the smaller species like minnow species, such as things like trout perch or spot-tail shiner. And unfortunately, gill netting is also quite invasive because the fish get stuck in the nets. They can get injured when you're trying to remove them from the nets, and some even end up passing away sometimes, which unfortunately it does happen. So the first objective of my project was trying to answer this one question here is, can eDNA detect the same, more or less species than gill netting? And so I looked at 7 species in total. So 4 of these species were larger bodied, such as walleye, sauger, yellow perch, and Coregonus species. So this includes the whitefish as well as Cisco species. I also had 3 smaller bodied species. So that's the rainbow smelt, spot-tail shiner, and trout perch. Walleye and sauger were included in this study because they're of high interest to a lot of Manitoba anglers. So those of you who love to fish, Lake Winnipeg has a ton of walleye and sauger, and so those were included in this study, especially for my next question, but I'll move on to that later. Trout perch was included because it tends to live kind of lower in the water column and tends to be missed by gill nets because of its size, and so I definitely wanted to include that one to see how often gill nets caught trout perch and if eDNA was able to detect them as well. And then I also included rainbow smelt here. Rainbow smelt is invasive to Lake Winnipeg. It was accidentally introduced, I believe, in like the 1990s. And so it's of special interest to the Manitoba government to keep track of whether or not this invasive species is still present. Unfortunately, they can have negative impacts on more native species because they compete for food as well as habitat. But they do have a natural predator. Walleye do love to eat them. And so I've included them in my study as well. All right, so here are my sampling sites of Lake Winnipeg. I had a total of 39 sites in 2022 and 21 sites in 2023 for a total of 60. And you can see they're spread out within the south and the north basin of Lake Winnipeg. So I just want to give you a quick breakdown of the eDNA method. It's definitely, there's so many steps, but this is kind of just a quick summary. Basically, you collect a water sample using a water bottle. Then that water is filtered through a filter paper using a vacuum pump. And then the DNA is extracted from that filter paper using a blood and tissue extraction kit. And then it's finally analyzed. For the analysis, for my project, I use qPCR, which allows you to bind synthetic sequences that are specific to the species that you're looking for. And then it's amplified millions of times through polymerase chain reaction cycles. Okay, so here are the results for my first question, which was, can eDNA detect more, less, or the same as gill nets? And so what I found was interesting. The numbers that you see in the table that I provide here, I've really simplified the table. There was a lot more data, but just for time, here it is. Here's a quick summary. Basically, the numbers are the total number of sites where that species was detected. So out of 60, a total of 60 sites, you can see that gillnets did seem to do a better job at catching the larger bodied species, so things like walleye, sauger, and yellow perch, than eDNA did at detecting them. But for the smaller bodied species like trout perch, spot-tail shiner, and rainbow smelt, we actually detected really high rates of eDNA at these sites, and a lot of the gillnets actually missed them, which was super interesting. So it does show that selectivity bias that you see with gillnets. And so this is important because, as you can see, even rainbow smelt, there was a total of 0 catches of rainbow smelt, but we did detect them at 19 sites across Lake Winnipeg. And so this is important because, as I mentioned, rainbow smelt is an invasive species in Lake Winnipeg. And if we only had the gill netting data, we may have concluded that they're no longer present in the lake, when in reality, eDNA was actually able to detect them. So here is my second question for my project. It was, can eDNA estimate the relative abundance of the species? So this is quite a controversial topic in the world of eDNA. And the ultimate question is, can we estimate how many fish were present at that sampling location? So here's just a calculation that fisheries management uses for calculating fish stock populations using gill nets. There's 2 different types. So there's the catch per unit effort and the biomass per unit effort. Catch per unit effort is the total number of fish caught divided by the total gill net soaking hours. And then the biomass per unit effort is the total fish weight divided by the soaking hours. And so these values is what I use to compare to my eDNA concentrations that I got from my qPCR analysis. But in order to see if there's a relationship between eDNA concentrations and gill netting abundance, I had to build a statistical model. So during my master's and through some awesome statistic workshops through FishCAST, I learned pretty quickly that things can get complicated with statistical models. So I'm not going to go over what my model included because I need a lot more time. There's a lot of variables, but I do want to share a really important step that may even affect your own projects and something that you might want to keep in mind. So when you're building your model, you need to check for something called spatial autocorrelation. And so I kind of give an example of this on my map, where you can see that I have 4 sites that are pretty close together. They're about 1 to 2 kilometers apart. And when I analyzed my data, I found that these sites actually shared very similar results. So to kind of get rid of that spatial autocorrelation, I decided to group my sites based on their proximity to one another. But unfortunately, when you group them, that leads into another issue that you have to deal with. And that's this concept that's called ecological fallacy. And there's a really good paper that explains it by Paulette et al. 2015. But basically, this concept is the assumption that data from an entire group is representative of individuals within that group, but that's not always the case. So taking a look at the graph, let's say that monkey A is one of my groups that I decided to lump together, all those nearby sites, and I call it group A. So now group A appears on the graph. You can see it seems to have a negative relationship between gill net, catch per unit effort, and eDNA concentrations. But that's not the case, because when you look at all of the data from all the groups together, you can see that there's actually a positive relationship. So that's the dotted line that runs across all 3 groups. And so this is what we call the ecological fallacy. And that's what you really need to consider when you're looking at the relationship between groups and within them. Okay, so here are my results for comparing my walleye eDNA concentrations to gillnet data. And it looks exactly like the graph from the ecological fallacy that I just showed you. At first, I got really excited because it did look like eDNA concentrations had a statistically significant positive relationship with gillnet CPUE and BPUE. So those are the 2 graphs with that I circled in red, because those were statistically significant, and you can see there's that line, trending line that goes upwards. But if you look at the two graphs at the bottom, the relationship, there is actually no relationship, no statistically significant relationship was found. It almost even looks like the trend is going down, that eDNA concentrations and gillnet actually goes down. But again, it wasn't statistically significant. And so the bottom line is that there does seem to be a positive relationship between eDNA and gillnetting abundance data, but only when you look at it at the entire lake as a whole. This means that we wouldn't be able to go to like a specific location, apply my model to estimate how many walleye were actually present at that specific site. But I do want to point out that I have very similar results, both for yellow perch and Coregonus species. So these are the larger bodied and more abundant species in Lake Winnipeg as well. And just trying to, if you were to try to model some of the less abundant species, things like spot-tailed shiner or rainbow smelt, it'd be very difficult to do because eDNA or gill netting data might be lacking for those less abundant species. All right, so, oh, sorry, I've skipped a slide there. Oh no, I didn't. Okay, so remember at the beginning, I mentioned that eDNA is a very sensitive technique where it only takes about one strand of DNA to detect a species. And because of this, contamination is super easy. When I collected my eDNA samples in 2022, I used the Manitoba government's fish stock monitoring boat and collected alongside the gill netting crew. But unfortunately, I ended up with a lot of contamination in my negative controls, which are included at every step of the eDNA process. These are basically water blanks that should contain no DNA, but if they do, it does indicate that somewhere along your process, you accidentally introduced DNA. At first, I was really panicking because I thought that all of my data from 2022 wouldn't be usable. And so I collected a second year of data in 2023 using a boat that was separate from the gill netting crew. And good news, my controls were all clean. So basically my message here is don't give up, just try again. Okay, so if I can give you a couple more pieces of advice, it would be to, one, attend as many conferences and workshops as you can. You never know who you might meet there, what kind of opportunities there might be. Two, apply for all the awards, even if you think you don't have a chance. And the third one, answer emails that may connect you to greater opportunities. And that's kind of vague, but this last one is actually how I got into my career in eDNA. I received a very random email from a government program here in Manitoba that connects graduate students to companies that might be looking for new employees. And the person I was in contact with just so happened to have met a company that did eDNA work here in Manitoba, which is very surprising because I considered eDNA to be a very niche field. And so I was very surprised that there was companies out there doing eDNA work. So that company that I work for, it is called Prairie Scientific. So I first started as an environmental technician in June 2024. And so, yeah, again, it did overlap with my master's. I was up late at night after work, basically doing my thesis up in Nunavut, which was, I mean, cool experience, but yeah, it was a little bit stressful for sure. But yeah, now I'm actually the eDNA lab manager for Prairie Scientific. So I got a promotion, woohoo. Yeah, and then so the company is based in Winnipeg, Manitoba, and we do environmental consulting on water quality and eDNA work. So I'm just going to talk a little bit first about our water quality program. This is mostly monitoring lakes in Nunavut for now, and we work in conjunction with the Kivalliq Inuit Association, which is an organization that represents the Inuit communities in the region. And some of the main concerns of the Inuit people is whether their water is safe to drink and the fish are safe to eat. And so what we do is collect water samples from lakes and streams that are of special interest to the communities and run metal analyses. We also collect tissue samples from fish, specifically lake trout, and test them for mercury, PCBs, and other toxic chemicals. We're also accompanied by elders during sampling trips, and we ask them a series of questions to gather Inuit traditional knowledge about the land, the water, and the living beings. And all of this data that we gather is publicly available. It's on DataStream if you were curious to know more about it and know more about what we collect. So now I work mostly on the eDNA side of things now that I'm a lab manager. And so the main projects I'm currently working on is creating an eDNA sampling protocol when collecting water in the Arctic regions. There's extreme cold weather conditions that you face when sampling up in Nunavut, which means that the protocols that typically work in the south, they don't tend to work up in the Arctic. And so I've been working on a field equipment decontamination protocol where I'm testing the use of bleach, heat, or both to get rid of DNA on our gear. So things like our water collection buckets and the auger to make holes in the ice. We added DNA from pink salmon, a fish species we know doesn't live in Nunavut Lake, to our field equipment, then used one of the 3 decontamination methods, and finally collected water samples under the ice. And samples are currently undergoing analysis using qPCR to see which of the 3 methods was most effective to remove that pink salmon DNA. So in the picture on the left here, that's me using a Tiger torch on our sampling equipment we used. And we used 2 sampling buckets. One of them has a really long wire you could kind of see in the picture, and that's used to kind of go further down into the water column. We also have an electric auger, that's the green machine laying on the ice there. That was up next to get decontaminated. And then on the right, I want to show the hole we cut with the auger to be able to collect our 3 water samples. And then you can see in the picture on the right as well, there's 3 bottles labeled torch 1, 2, and 3. And those are the containers where we pour our eDNA samples into. So we're testing the lake water samples for not only pink salmon, but also lake sturgeon as well. Nunavut Lake is well known to have lake sturgeon. And we want to make sure that we test the water samples for their DNA as well, because if they don't show up in our water samples, that might mean that our decontamination is actually ruining some of that DNA that might be collected in the water samples. Okay, we're also going to various lakes in Nunavut collecting eDNA samples to create a baseline data on the fish communities that are currently present in the Arctic. This is because of warming of water systems here in the south. There seems to be some fish species that are migrating to cooler water systems, and they're getting pretty close to the Arctic. And now, if they're moving further north, this can have a lot of negative impacts for species that already live there, where their habitats can get lost or destroyed, and there may be higher competition for food. In my master's project, I talked about using qPCR to analyze data from a single species, but there is another method that I used. This is called metabarcoding, and essentially it allows you to look at the entire fish community as a whole instead of one at a time. So that was pretty cool. I got to learn all about qPCR in my master's, and then into my career, I got to learn a whole different side of the eDNA technique, which was very cool. Now, the other thing that I'm working on is getting our lab accredited for eDNA work. And essentially what accreditation is, it's having an official designation from a third-party organization that the results and the procedures that you use in your lab are accurate and consistent. And so this would tell your clients that the results you provide are credible. And the lab would be regularly evaluated through audits and tested against other labs through things like proficiency tests or internal tests. And this evaluation is based on an international standard that's written out, and that standard is called ISO 17025. And currently, there's actually very few eDNA labs globally that are accredited. I know there is one in Canada. I believe it's Fisheries and Oceans Canada has one in New Brunswick. But that's the only one, I believe, in Canada that is accredited. There's a couple overseas, but yeah. Okay, so just a few key points that I wanted to mention before I conclude today. And that is one, go outside of your comfort zone and take those opportunities. Number two, science, it doesn't always work, and that's okay. And I'm still learning that even in my career, and it'll keep happening. You're always learning something new every day. And yeah, you just gotta keep trying. And then the third thing is networking and reaching out is really important to your future career. And so if you're curious to learn more about Prairie Scientific, the work we do, and the services we provide, feel free to contact me by email, or I have the link to our website. You can also follow us on LinkedIn. There isn't anything on there right now. We're going to be posting our first post next week. And so we're still working on that. But yeah, feel free to follow us on LinkedIn. And thank you for joining me today. And I'd be happy to answer any questions you might have. Thank you. Thanks, everyone.